Teno Virus 7 was identified from 2 of the 11 patients with Kawasaki disease, but none of the 22 matched control subjects in a prospective etiology hunt. I'd like to talk about our recent research into the etiology of Kawasaki disease. In Japan, 16,000 children develop Kawasaki disease every year. To date, 400,000 Japanese children have suffered from this illness. However, the etiology of Kawasaki disease has not been revealed yet. Our epidemiological analysis suggested that the main etiological agent of Kawasaki disease in Japan may be transmitted from a child to a child, most likely through cough, feces, or direct contact. We predicted that this agent is a very common virus and infects most of the Japanese children. This virus may infect children persistently. Meanwhile, the metagenomic sequencing by a next-generation sequencer coupled with a bioinformatical analysis emerged as a methodology to detect known and unknown microbes. We conducted a pilot study to identify the etiological agent of Kawasaki disease using metagenomic sequencing and bioinformatical analysis. This is the scheme for a study at hospital. We defined 3 groups, which are the group of Kawasaki disease patients and 2 groups of control subjects. One control group was children with diarrhea, and the other control group was patients with respiratory infections. Control subjects were matched to the Kawasaki disease patients in terms of age, gender, and the date. Serum, whole blood, nasopharyngeal aspirate, and feces were collected from patients. DNA and RNA are extracted from these samples, and RNA was reverse transcribed into cDNA. These DNAs were sequenced. We detected all the microbes, including virus, bacteria, fungi, and archaea, in the specimens by bioinformatical analysis. We enrolled 11 Kawasaki disease patients in 1-year period. To each patient of Kawasaki disease, 1 control with diarrhea and 1 control with respiratory infection were enrolled. Control subjects were matched to the Kawasaki disease patient in terms of sex, age, and date. We developed a database-driven software which matches a Kawasaki disease patient to controls. We determined the host genotypes of each participant in terms of the known risk alleles for Kawasaki disease. We selected ITP-KC and BLK as the risk alleles. The per capita number of the risk alleles are comparable between Kawasaki disease patients and the control subjects. Samples were pooled in each of the 3 groups, which are Kawasaki disease, diarrhea, and respiratory infections. We chose conditions which are favorable for viruses. These conditions are size filter and multiple displacement amplification. Nuclease was applied to some samples to eliminate human and bacterial DNAs. DNA libraries were prepared as per manufacturer's instructions and analyzed using an Illumina sequencer. Bioinformatical analyses were performed mainly through Livermore Metagenomics Analysis Toolkit, or LMAT. LMAT classifies each of the billions reads down to the species level. This is the result from LMAT. A unique feature of the serum and whole blood samples collected from Kawasaki disease patients was that viral reads were more abundant than bacterial reads. For example, in the whole blood DNA from Kawasaki disease, 95% of the reads were viral. LMAT classified the reads down to the species level, and this is the composition of viral reads, or virome. In all of the samples pooled from Kawasaki disease, the dominant viral species was always Teno Virus 7. For example, in the blood DNA pooled from Kawasaki disease, more than 98% of the viral reads was Teno Virus 7. We then applied PCR and Sanger sequencing to each of the 33 individual samples to detect and sequence Teno Virus 7 respectively. Teno Virus 7 was detected in 2 Kawasaki disease patients, but was absent in all of the 22 control subjects. The upper vertical lines indicate differences between the sequenced and reference DNA. The lower lines, arrowhead, indicate the differences in the amino acid sequences. These 2 Teno Virus 7 isolated from Kawasaki disease patient 1 and 5 had identical amino acid sequences. This variant was not present in GenBank, European Molecular Biology Laboratory, or DNA Data Bank of Japan. This table shows the characteristics of 2 Kawasaki disease patients who were positive with Teno Virus 7. Both patients were categorized as having high risk for unresponsiveness to intravenous immunoglobulin. Both patients developed transient coronary aneurysms. We evaluated the correlation between Kawasaki disease and Teno Virus 7 or host genotypes. The correlation between Kawasaki disease and Teno Virus 7 was not significant, with an odds ratio of 4.83 and a p-value of 0.111. However, this was comparable to the correlation between Kawasaki disease and susceptible genotypes. For example, the correlation between Kawasaki disease and the risk allele of ITPKC was an odds ratio of 3.82 and a p-value of 0.161. The pathogenicity of Teno virus is unknown. Teno virus is highly infectious, and Teno virus is found everywhere in any biological samples, for example, blood, sweat, sewage. Almost all human beings are infected with at least one or more of 29 Teno virus species. All Teno virus species persist throughout the host lifespan. Teno Virus 7 was reported in 2000 by Allet. A small pilot study identified a new variant of Teno Virus 7 from 2 patients of Kawasaki disease but from none of the control subjects. The correlation between Kawasaki disease and Teno Virus 7 was not significant, with an odds ratio of 4.83 and a p-value of 0.111. However, Teno virus are ubiquitous and infect the host persistently, which is consistent with the epidemiological prediction for the causative agent of Kawasaki disease. These are my collaborators. Thank you very much.